errors in splicing are actually can play an important role in human disease. And in the cases where disease is caused by single nucleotide polymorphisms or SNPs, these are just single base pair changes in the DNA sequence. Of these cases of these diseases where SNPs are the cause, 15% of them affect splicing specifically. And so one example is hemophilia, which we mentioned when we were talking about human pedigrees as being prevalent in the royal families of Europe, and probably the most well-known case is of Alexis, son of Nicholas II, Tsar of Russia, who suffered immensely from this condition. And hemophilia affects a clotting factor that is responsible for blood clotting, and a mutation within an intron affected the splicing of exons such that a premature stop was produced. And so his cells produced only truncated protein that was non-functional. And so as a result, he had this condition where his blood wouldn't clot. So splicing is important. In some organisms, particularly in the nematode C. elegans, some organisms can actually engage in trance-splicing. And what this means is that normally we see splicing happening where we have a single transcript from a single gene where the introns are getting spliced out and the exons are getting spliced together. C. elegans can take this one step further, where it can take transcripts from different genes and splice exons from different genes together to create a new protein. So that's kind of cool. Not too many organisms do this, but one happens to be a genetic model organism. So another interesting variant of the splicing that we were talking about is self-splicing that does not require a spliceosome. And these are introns. As you can see, they can be quite long, that form, where their sequences form secondary structures. And these secondary structures are often there's a pattern to them, such that there's a certain number of stem loop structures that characterize different types of self-splicing introns in group 1 or group 2. And the structure of these secondary structures influence the self-splicing ability of these introns. All right, so we're going to talk about alternative processing in more general terms, and alternative processing just means that the same pre-mRNA can be processed in different ways to yield ultimately different proteins. And so one form of alternative processing is alternative splicing, where let's say we have 3 different exons in blue, yellow, and red, and one mature mRNA can be spliced, one pre-mRNA can be spliced to yield a mature mRNA that includes all 3 exons, whereas another splice variant that is spliced differently might yield a mature mRNA that only includes exon 1 and 3. So that's one example of alternative splicing. Another form of alternative processing has to do with the cleavage site. And so recall during the last lecture on transcription, the pre-mRNA, as it's being transcribed, gets cleaved, and this is an important part of the transcription termination process. So this cleavage site can actually vary. So you can have alternative 3' cleavage sites, and that might occur. So here in this pre-mRNA, there's a possible cleavage site here that's cleavage site 1, and then there's a possible cleavage site here that's cleavage site 2. And so cleavage site 1 would yield this version of exon 2, in the mature mRNA, whereas cleavage site 2 would yield this longer version of exon 2. And so different cleavage sites can give you different mature mRNAs and ultimately proteins that vary in length and therefore structure and therefore function. Okay, so one example, and let me just take a second to, all right, I just wanted to fix the writing there so that you could actually see it. Okay, so we've got a gene in our genome that is, you know, this is just a schematic of it. It probably has more exons than this, maybe. And it can be the pre-mRNA can be alternatively processed such that you can have different combination of exons and also different 3' cleavage sites. So these two different mature mRNAs result because of both alternative splicing and alternative 3' cleavage sites. So this mRNA produces a protein called calcitonin that's expressed in thyroid, and in thyroid it regulates calcium levels. This same gene, alternatively processed, can produce a different variant in brain that is called calcitonin gene-related peptide, or CGRP, and it actually serves a very different function in controlling blood vessel dilation and also regulating pain. And there's some indication that this particular protein is implicated in migraine headaches. So, same gene, very different functions. When different transcripts are produced from the same gene that yield different proteins, these are called isoforms. Okay, clicker question. Alternative 3' cleavage sites result in A, multiple genes of different lengths, B, multiple pre-mRNAs of different lengths, C, multiple mRNAs of different lengths, or D, all of the above. So what this question is asking is really about when does the 3' cleavage happen, and how does that affect the DNA, pre-mRNA or mature mRNA? And the correct answer is that it produces multiple mRNAs of different lengths, or C. The genes are not going to change as a result of the different cleavage sites, and the pre-mRNAs will actually, though I guess you could also argue that different cleavage sites will also affect the pre-mRNAs and make them be of different lengths. That's kind of an interesting, let me just check this out for a second because, all right, so clarification. When I showed you this cleavage site, this is actually not the cleavage site that we're talking about. I misled you. I apologize. So there's two cleavage sites. So there's a cleavage site that occurs during transcription, essentially before it's terminated, and then there's additional cleavage that happens. So here we've already been cleaved in that figure that I showed you. This is another cleavage site. This is different. And so actually the pre-mRNA, and we see this in this figure. So here's the pre-mRNA. It's the same length, but the cleavage sites here are different. All right, so there's a cleavage site that occurs that's important for transcriptional termination, but then there are other cleavage sites that that can exist within the pre-mRNA that influence the ultimate, that are important in mRNA processing, and that influence the final size of the mature mRNA. So to clarify that. All right, so moving on. RNA editing is another form of RNA processing, and this kind of is a little bit mind-blowing because you start out, we think that gene sequence influences mRNA sequence directly, and then that should so the central dogma, right? DNA sequence to RNA sequence to protein sequence. This kind of messes with that a little bit. So here we have our pre-mRNA. We have a guide RNA that comes up and anneals and interacts with the mRNA. But the guide RNA has some extra nucleotides in here, and these nucleotides kind of get looped out in the annealing process. Then the guide RNA is used as a template for the addition of extra nucleotides within the mRNA that are then change the protein structure. Okay. This is we think it's incredibly rare in mammals. In Drosophila, it occurs about 3% of the time, we think. There was actually recently, and I put a link to this Popular Science, PopSci article on Blackboard that was about a squid, the long fin shore squid, something like that, was recently discovered to have about 60% of its RNA transcripts are edited, which is totally crazy. And so, we still need those results to be verified and repeated, but that's what how it stands right now. You may hear from me. Well, you may hear at some time later that maybe that's not exactly right, but it's pretty interesting. Okay, so that's RNA editing. Just to got rid of some extra text there, sorry. Just to kind of go over this again. Here is the DNA structure with our promoter upstream of our transcribed region, including exon 1, intron, exon 2, intron, exon 3, and an intron. Okay, that's again indicating that endotranscription ends with introns, which is not generally true. Anyway, so we get our pre-mRNA, that's just transcribed directly from the gene. We get our 5' cap. We get our cleavage here. We get our poly A tail. And then we get our introns are spliced out. So this is just a schematic summary of what happens during RNA processing. All right, I am now going to get into the final part of the lecture, which talks about the different types of RNAs, some of the major ones, and how they might be alternatively processed. And so I'm going to start out with the transfer RNAs, or tRNAs, and these are the RNAs that are responsible for bringing amino acids to the ribosome during translation. So tRNAs have a characteristic cloverleaf structure, and each tRNA can carry only one type of amino acid. So here are different schematic models. So this is what do they call it? A ball and stem something molecular model of the protein. Here's a different type of model that shows that three-dimensional structure. And here RNA, just to remind you, this is an RNA. It's single-stranded, and it has complementary pairing on the stems, and this helps create a secondary structure that is three-dimensional that helps it accomplish its function. So, okay. So just real quick, there's an arm down here that is the anticodon arm that interacts with the mRNA during translation. There is a couple arms here, and then this is the site where the amino acid is carried.